In confined commercial poultry houses, thousands of birds share a single environment. These intensive rearing conditions create an environment where concentrations of airborne viruses, bacteria, and fungi reach extreme levels. Without a highly specialized cellular defense network, a commercial flock's lifespan would be exceptionally short. A bird's survival relies entirely on an immune system capable of blocking infection and halting microbial replication. The overall organization of this avian defense network shares a common baseline with mammals. Both systems rely on a dichotomy between T cells, which mediate attacks, and B cells, which produce antibodies. But the anatomical tools used to generate these cells diverge sharply. While mammalian B cells mature in the bone marrow, avian B cells develop in a unique sac-like extension of the hindgut called the bursa of Fabricius. These distinct biological mechanics dictate how a bird responds to infection. Mapping exactly where avian immunity deviates from the mammalian model is required to develop effective vaccines and engineer long-term disease resistance in global poultry populations. When pathogens breach physical barriers, the innate immune system drives the immediate response. As shown in this layout, local macrophages form the first line of defense against viral particles. Using surface toll-like receptors, they bind invading pathogens, signaling cells to secrete cytokines, triggering localized inflammation. This cellular response is supported by avian natural killer cells. Highly concentrated in the intestinal epithelium, these large granular lymphocytes express unique CD8 alpha-alpha homodimers. This allows them to identify and lyse virus-infected target cells without requiring prior antigen presentation. Innate immunity is further backed by a protein-based complement system circulating in the plasma. However, the architecture of avian complement operates quite differently than the standard mammalian pathways. In mammals, the classical complement pathway is dominant, but in chickens, classical activity is difficult to demonstrate. Instead, birds rely heavily on the alternate complement pathway. Unlike the mammalian version, the avian alternate pathway is directly activated by circulating antibodies. This unique activation route allows the alternate pathway to rapidly bind to and destroy extracellular bacteria. It provides an essential stopgap to control bacterial replication during the days it takes for the adaptive immune system to fully mobilize. Once the adaptive immune system mobilizes, T cells take over. For a T cell to recognize a foreign pathogen, the antigen must first be broken down by a macrophage and presented on the cell surface by a glycoprotein receptor known as the major histocompatibility complex. The mammalian MHC contains over 200 genes, but the chicken B locus is severely compressed to 19. It is strictly divided. The BF region encodes class I molecules on all nucleated cells. The BL region encodes class II molecules, restricted to antigen-presenting cells. The T cells interacting with these receptors also present a distinct cellular anomaly. Chickens possess an unusually high concentration of gamma-delta T cells, which can make up 30 to 50% of all circulating lymphocytes. When T helper cells encounter presented antigens, they polarize into Th1 or Th2 effector cells. This differentiation is driven by an expanding specific network of avian cytokines, including interleukins 12 and 18. Because the 19-gene B locus provides so few options for antigen presentation, a flock's ability to recognize a specific virus is strictly bound to its genetics. A single variation in an MHC haplotype often dictates whether an individual bird will succumb to an infection or show zero phenotypic signs of disease. While T cells clear infected tissues, the bursa of Fabricius produces the B cells responsible for humoral immunity. These cells secrete the antibodies that neutralize pathogens in the bloodstream. The chicken humoral response utilizes three primary classes of immunoglobulins. Like mammals, birds produce IgM first and use IgA for mucosal defense. Their primary circulating antibody operates differently. Compared to 4-domain mammalian IgG, the avian equivalent, IgY, features 5 heavy chain domains and lacks a flexible hinge. Cloning data suggests this rigid IgY is the ancestral precursor that eventually split into the IgG and IgE isotypes found in modern mammals. The absence of a hinge region changes the physical mechanics of how these molecules bind to viral targets. Because avian antibodies interact with foreign proteins so differently, veterinary immunologists must use entirely specialized diagnostic tests to measure flock immunity accurately. Every environmental pathogen presents a unique antigenic shape that the immune system must recognize and match. Yet the avian genome is built on a highly restricted, heavily bottlenecked set of immunoglobulin genes. To reach the necessary level of antibody diversity, chickens cannot rely on the extensive VDJ recombination process used by mammals. Instead, birds utilize a distinct genetic workaround known as somatic gene conversion during B-cell maturation in the bursa. The rearranged light and heavy chain genes acquire massive diversity by copying short sequences from a cluster of homologous pseudogenes located upstream and splicing them into the active gene. This process of gene conversion allows birds to match the mammalian adaptive repertoire despite their severe genetic bottlenecks, serving as a critical evolutionary workaround for pathogen defense. When a chick hatches, its own adaptive immune system is completely naive. To survive early exposure to housing environments, the chick relies entirely on a reserve of these uniquely generated antibodies passed down directly from the hen. Immunity transfer is partitioned during egg formation. IgY accumulates in the yolk sac, while IgA and IgM enter the amniotic fluid. As tracked on this graph, these maternal antibodies flood the embryo's circulation, peaking 2 to 3 days post-hatch, before beginning a linear decline. While this passive immunity keeps young chicks alive, it creates a serious bottleneck for commercial production. If a live vaccine is administered in ovo or immediately at hatch, pre-existing maternal antibodies will rapidly neutralize the vaccine antigen, providing negative feedback that prevents the chick from developing its own active immunity. To manage this, the poultry industry continuously profiles flock immunity using precise in vitro assays. Researchers measure defense capacity using chromium-51 release assays to quantify natural killer cell activity, or calculate macrophage activation by testing for nitric oxide-inducing factors. Mapping these exact antibody degradation curves and cellular baselines is critical. It allows producers to time their vaccination schedules perfectly, ensuring a live vaccine takes effect exactly as maternal protection fades. The biology of the chicken proves that there is more than one way to construct an immune system. Birds face the exact same high-density pathogen pressures as mammals, but they counter the threat utilizing a highly distinct evolutionary toolkit. Their defense relies on an ultra-compact MHC B locus to present antigens, the ancestral IgY structure diversified through rapid somatic gene conversion, and a highly partitioned system of maternal antibody transfer. Today, these biological mechanics are directly leveraged by the commercial poultry industry. Because the B locus is so strictly linked to antigen presentation, modern breeding programs can actively sequence and select for the specific MHC haplotypes that generate superior disease resistance. Mapping these distinct host factors is a practical necessity. It provides the biological blueprint required to engineer resilient flocks, design precise vaccines, and secure global commercial poultry production against an evolving microbial environment.